thank you.
Don't forget to write your questions down.
Yeah, no, definitely. And so, it's great to be here. Thanks to the organizers of this symposium, and thanks to all of you for coming. I'm obviously a psychiatrist and a brain scientist, and this is all incredibly important and exciting to me. But I also, it's great to get out of the laboratory and get feedback and questions and hear your thoughts on what we're doing. That's really always invigorating and exciting. So I hope this is the start of a conversation during the question period and thereafter, and I look forward to hearing all of those. So I'm going to tell you a story about work in my laboratory, and it's really about cells in the brain, how they generate different patterns of electrical activity, and why that might be relevant to the way that we think and the way that thinking can become disordered in disorders such as schizophrenia. And don't worry about the details. I'm going to talk about a lot of details that you can just totally ignore and forget about. But I think the big pictures hopefully will give you a glimpse of where brain science is really going and why it really is relevant and hopefully exciting in the ways that we think about mental illness and especially treating mental illness in the future. So, Brandon already did a fantastic job of emphasizing why cognition is such an important issue in disorders such as schizophrenia. It's really the thing that's holding people back from leading the lives that they really want to lead, and it's the thing which right now we have the most limited treatments available for. And so we really want to understand what in the brain is driving cognitive deficits in conditions such as schizophrenia. And of course, this is challenging because the brain is really complicated. And this is a picture from the Brainbow project at Harvard. And here they've colored, lots of different neurons, lots of different colors. And when they do that, you can see the incredible diversity of neurons in the brain. There's so many different types of neurons, and they seem to have very special shapes, and that implies that maybe they have very special functions. And how can we possibly understand what all these functions are and how they interact to cause mental illness? And so that's the story that I'm going to tell you today. How can we try to figure out what specific cells in this mosaic are abnormal in disorders such as schizophrenia? And if we can figure out which ones are maybe at the heart of some of these issues, then what do they do? And if we can figure that out, how do we fix them in order to kind of address some of these major issues such as cognitive deficits in the setting of mental illness? So I'm going to start by telling you about specific types of cells in the brain, and this is the first super jargony term I'm going to throw out there. I'm going to tell you about parvalbumin interneurons. And it's unfair that they have such a complicated name, but interneuron means that they're just a type of cell in the brain that when they communicate with other cells in the brain, they tend to inhibit their activity. So kind of the same way that a conductor tells different instruments when to get louder or when to get more quiet, these cells conduct the activity of other cells around them, and they tell them when to pause and when to become active. And these are a particular type of inhibitory neuron that can be recognized because they have a protein in them called parvalbumin. So we call them parvalbumin interneurons. All right, so what do parvalbumin interneurons do, and why might they be abnormal in schizophrenia? Well, it turns out there's a lot of evidence looking at tissue from individuals who had schizophrenia suggesting there is something wrong with these parvalbumin interneurons. And so what I wanted to do in my laboratory was actually show that, hey, this isn't just some random change, that problems with these neurons actually can cause some of the major features of schizophrenia and can help us to understand this disorder. And so we studied mice. We studied mice that had abnormal parvalbumin interneurons. And so here's an example of electrical activity from a normal parvalbumin interneuron and from a mouse with abnormal parvalbumin interneurons. And you see all these wiggles. So on the top are wiggles from a normal parvalbumin interneuron. Each of these wiggles, we call them an action potential. And these are the electrical signals that one brain cell uses to communicate with another brain cell. And on the bottom are the electrical signals from a mutant parvalbumin interneuron that has some abnormal developmental properties. And I think what you can appreciate, even someone who is looking at this kind of thing for the first time, hey, on the top, there's these wiggles, they go on, they happen at a fast rate, and they're pretty stable over time. And what that means is that this neuron is able to communicate at a very, very high rate and do so, very, very efficiently over time. And on the bottom, yeah, there's a parvalbumin interneuron. It's generating all of these action potentials that kind of seem to run down or poop out over time, and maybe that affects the way that these neurons would communicate with other cells in the brain. And so we can actually measure that. We can actually measure the output from parvalbumin interneurons onto other cells in the brain. And again, here I've shown the normal output in black and output from a mutant cell in blue. And what you can see is, well, there's less output from the mutant cell. So in these mice, these parvalbumin interneurons, they're not working as well, and they're not able to communicate with other cells in the brain as well, okay? So that's the first part of the story. There's something wrong with parvalbumin interneurons in schizophrenia, and we're looking at mice that have similar abnormalities to find out what are the consequences of these problems. And so what are the consequences? Why does it matter if this one, I already told you there's all these different cells in the brain, so who cares if there's something wrong with one particular cell type? Well, we actually know a little bit about what these parvalbumin interneurons do. They actually, as I said before, control patterns of electrical activity in the brain. And so we can actually record patterns of electrical activity from normal mice and from these mutant mice which have problems with their parvalbumin interneurons. And again, it just looks like a bunch of wiggles. And we actually care a lot about these different wiggles. We can actually measure wiggles of different types. So there's some slow wiggles and there's some really fast wiggles. And these correspond to rhythmic activity at different frequencies. When you hear a song, it's the same thing. There's some really fast parts and some really slow parts. And we can basically measure the size of the fast and the slow parts, and we call these oscillations. So there's some slow oscillations at low frequencies. There's some fast oscillations at high frequencies, and we can measure the size of these different oscillations. And why is this important? Well, these parvalbumin interneurons generate fast frequency oscillations, these fast wiggles in those electrical signals. And we call those gamma oscillations. And these gamma oscillations, they're actually important. You can actually measure them even from the brains of all of us sitting in this room if you just put an EEG electrode on the outside of your skull. And these gamma oscillations tend to appear whenever you're doing something difficult, when you're concentrating, when you're paying attention, when you're trying to remember something. And the same is true of these mice. We can measure the changes in the size of these different electrical patterns of activity, the change in the size of these wiggles. And these fast wiggles, the ones at high frequencies, between about 30 and 100 hertz, they go up a lot when we have mice doing what are challenging cognitive tasks for mice, when they learn a rule and they have to shift from that rule to another rule, or when we have the mice socially interacting with another mouse. Okay, and so what I've shown you here in black are the size of the gamma oscillations in normal mice. And so if we look at these mice, these mutant mice that have broken parvalbumin interneurons, their gamma oscillations are smaller. They just can't seem to rev up those fast electrical wiggles as well as normal mice. And so now you're all thinking, well, I've told you about two totally irrelevant things. I told you about parvalbumin interneurons, and now I told you about gamma oscillations. Well, it turns out that these gamma oscillations are also abnormal in individuals with schizophrenia. There too, when people are doing tasks where they have to switch what they're paying attention to or really concentrate and remember information, those gamma oscillations don't get revved up quite as well. And so why does this matter? Why are we studying this in so much detail? Well, we think these gamma oscillations actually are important for carrying out those cognitive tasks. And we were able to show that in these mice. So we have these mice doing a task which is really hard for the mice. They have to dig in bowls to find food, and the different bowls have different odors. So one bowl might smell like garlic, and another might smell like coriander, and they're filled with different things. Some are filled with sand, and some are filled with wood chip. And mice love to dig in these bowls to find food, but of course we make it tricky. Only one bowl has food, and there's a certain cue, like the smell of garlic, that'll tell the mouse, hey, this is where the food is. And mice learn that, and then, because we're scientists and we don't make things easy for the mice, then we switch it up, and then it changes. So it's no longer the smell of garlic, it's now the presence of sand in a bowl that tells a mouse where to find food. Okay, and so we can measure how well the normal mice and the mice with broken parvalbumin interneurons do on this sort of a task. And here's a graph, think of it as like a report card for these mice. So we measure the number of trials it takes them to learn the rule, and we measure basically the number of trials it takes them to learn the first rule, and then a shift to a new rule. And in black is the data for normal mice, and in blue is the data for these mutant mice. And what you can see is that the mutant mice, they can learn the first rule just fine. But when you have them switch to a second rule, it's really, really hard. It's really hard for them to switch from paying attention to a smell like garlic to now paying attention to a texture like sand. And this is actually the same kind of deficit that's seen in many disorders such as schizophrenia. It becomes just unduly challenging sometimes to pay attention to these complex cognitive kind of switches. And so in these mice, simply by disrupting the function of parvalbumin interneurons, we're able to recapitulate and show that they show these same sorts of cognitive deficits. Now, from a scientific standpoint, this is really exciting for us because, as I said before, we know that these parvalbumin interneurons are broken in schizophrenia, and now we've shown that that matters. But for all of you in the audience, you want to know, well, okay, that's great. Yeah, you've maybe shown that thing A causes thing B, but what we want to know is, can you fix it, right? If we know what's wrong with these parvalbumin interneurons, can we fix it, and would that actually fix the ability to switch from one rule to another? Okay, so I've told you that the brain is made up of all these different types of cells, and that there's one particular cell type, these parvalbumin interneurons, maybe shown here in yellow, that we think are really important. So it turns out we can fix them, and we use cool new technologies, some of which were developed and really pioneered at Stanford, where I studied before I started my laboratory at UCSF. And these new technologies allow us to turn on just the yellow cells, just the parvalbumin interneurons, with light. Okay, so we can actually genetically engineer mice so that their parvalbumin interneurons, these broken cells, become sensitive to light. And when we shine blue light, those parvalbumin cells will wake up and they'll become active. And this is great because we can actually flash blue light at different frequencies. So we can deliver fast flashes of blue light at gamma frequencies, and this will actually wake up those parvalbumin interneurons and bring back the gamma oscillations. And we can see whether or not when we do that, that actually improves the behavior of our mutant mice in ways that might be relevant to treating schizophrenia. So we can do this actually in behaving mice. So this seems really, it seems totally far out, but this is the kind of thing that's really revolutionizing brain science, is we can actually implant a tiny little fiber optic cable into the brain of the mouse and deliver light, and now the mouse has been genetically engineered, so when we do that, we wake up the parvalbumin interneurons and turn on gamma oscillations. So what happens when we do this? Well, here's the kind of experiment we do. On day one, we have a bunch of these mutant mice. They learn an initial association. They learn a rule shift. We already know they're bad at that. So on day two, we have them do the same thing, but for some of the mice, we give them flashes of blue light to activate the parvalbumin interneurons and restore the gamma oscillations. And we see what happens to the rule shift when we do that. Do they do better? And then on the third day, we turn off all the light and we test them again, just sort of everything back to normal, and see how the mice do. So we have two groups of mice in this. We have one group shown in white. We always have a control group in science. That's the unlucky control group. They don't get any effective light stimulation. And then in yellow, we have the mice that are getting this light stimulation. Their parvalbumin interneurons are getting activated. We're generating gamma oscillations. And what you can see by looking at this graph is that on day one, when there's no light stimulation, all the mice have a really hard time learning the rule shift. They take many, many trials to learn. But on day two, the yellow bar, the mice that get the stimulation, we wake up their parvalbumin interneurons, we generate gamma oscillations. Those mice actually perform just like normal mice. They actually have no trouble with this rule shift. It's all of a sudden is easy for them. And what's amazing is that when we turn off the light and we repeat this task again on day three, the mice that the day before we gave them this stimulation and we generate gamma oscillations, they continue to do well. In fact, we can test them a week later and they still do well. It's as if they had a eureka moment, kind of hearkening back to what Brandon was talking about. It's sort of like we did brain training for these mice, but by giving them this stimulation, we turbocharge the brain training. It happens really, really fast for these mice, and all of a sudden they get what this task is all about, and it's not hard for them anymore. So the moral of the story here is that cells generate patterns of brain activity which drive behavior, and we can fix behavior. But in order to do so, we had to understand what were the patterns of activity that drive those behaviors normally, and how are they altered in disease. It turns out if we use other patterns of light stimulation and we don't drive gamma oscillations, we do something else, it doesn't work. We had to drive the gamma oscillations in order to restore cognition in these mice. And so I just want to end with this slide just to show where we're going. We're thinking obviously now about the human brain. And so this is actually a movie of activity from many different locations and at many different frequencies in the human brain. And what I want you to see here is just, it's not random. There are patterns here. You see these clouds that appear or disappear in a coordinated way. And so maybe by understanding these patterns, we can understand the patterns that are associated with feeling more depressed or more anxious, or the patterns that are associated with feeling more hopeful and more optimistic and being able to pay attention and being able to learn. And we can think of new ways to enhance these. And so that's it.